Millions of dollars and, honestly, thousands of GPU hours are currently being poured into training these massive AI foundation models to predict human biology.
Yeah, just an unbelievable amount of resources.
Right, and yet a rudimentary mathematical average is frequently beating them. Welcome to the Deep Dive. Today we are looking at a breakthrough paper that proves bigger isn't always better in computational biology, especially when we are trying to predict how a cell will react to genetic alterations.
We are dissecting a new framework called DeSCOPE, that stands for Decoding Single-Cell Omics of Perturbation Responses. And it targets one of the most stubborn bottlenecks in biomedical research.
Because we are essentially trying to build what you could call a virtual cell, right? Like a perfect digital twin for you, the listener, that allows us to run millions of genetic experiments entirely in software.
Exactly. But predicting the cascading effects of a genetic tweak is, well, it's mathematically brutal.
It is. And we have an incredibly dense map of information to navigate today. We are going to look at the underlying architecture of DeSCOPE, how it handles predicting the effects of genes it has never seen, how it transfers that knowledge to entirely new human tissues, and finally, how it predicts complex double-gene combinations while mapping the physical accessibility of our DNA.
It's a lot to cover, but it really reshapes the field.
Okay, let's unpack this. We all know how CRISPR works in a physical lab, right? Knocking out a specific gene is pretty straightforward at this point.
Yeah, the physical edit is the easy part.
Exactly. The bottleneck isn't the edit itself. It is the cascading biological noise. You knock out one transcription factor, and suddenly the cell is scrambling hundreds of downstream pathways just to compensate. Modeling that mathematically across the entire transcriptome is a total nightmare.
It really is.
Before we get into the algorithm, ask yourself this. If you had a fully functioning digital twin of your own cellular network sitting on a hard drive, what is the first intervention you would want to simulate?
Oh man, that completely reframes how we think about disease treatment, doesn't it? But, to understand why simulating this is so difficult, we need to look at the baseline problem. The data we use is cRNAs, single-cell RNA sequencing.
And for anyone who needs a quick visualization, how should we picture that?
Think of it like reading the active recipe book of a single cell. It provides a highly dimensional snapshot of every single RNA transcript a cell is actively trying to cook up into a protein.
So if your RNA-seq is the recipe book, a CRISPR perturbation is like taking a giant black marker and just completely crossing out one specific recipe in that book.
That's a perfect analogy. And we want to know how the rest of the cell reacts. Like, if it can't make that one recipe, does it start overproducing 5 others to compensate?
Right. And to predict that, the field has leaned heavily into these massive AI foundation models, right?
Yeah.
Systems like GEARS, STATE, or SCGPT.
Yeah, which are built on transformer architectures with millions, sometimes billions of parameters.
But what stands out in this paper is the sheer inefficiency of those massive models.
Right.
Like, they frequently fail to beat a baseline calculation called perturb mean.
Yeah, that part is wild.
I mean, perturb mean is literally just taking the mathematical average of the cellular reactions we have already observed in the training data. The fact that basic averaging outperforms multi-million-dollar neural networks, it suggests these giant models are severely overfitting.
Completely. They get totally lost in the noise of single-cell data because they lack the right biological constraints. And that is the exact problem DeSCOPE solves. Instead of a massive transformer, it relies on a conditional variational autoencoder, or CVAE.
Which is much smaller, right?
Exactly. It is a much more lightweight, elegantly constrained architecture built using simple multi-layer perceptrons.
Let's visualize the computational workflow of that CVAE. You can think of it as a highly sophisticated zip file program.
I like that, a compression algorithm.
Yeah. The first component, the encoder, takes the massive, chaotic, high-dimensional cRNA-seq data of a cell and compresses it down into a tight mathematical summary. We call this the latent space.
Right, it strips away the noise.
Exactly. It captures the fundamental state of the cell. Then, the decoder takes that compressed latent space and attempts to decompress it back into a prediction of the new, perturbed cellular state.
But the genius of DeSCOPE isn't just the compression. I mean, compression is standard. It is the biological prior it injects into the system. DeSCOPE utilizes ESM2 embeddings.
ESM2, that's the Meta AI model, right?
Yeah, it's a 15 billion parameter protein language model developed by Meta. DeSCOPE takes this model and generates a 5,120 dimensional mathematical vector, essentially an incredibly detailed ID card for every single gene based entirely on its protein structure.
Now, wait. I have to push back on the causality there. ESM2 is trained strictly on reading protein sequences. But RNA expression and protein folding are two entirely different steps in the central dogma of biology. How does feeding a model an ID card based on protein math help it predict upstream RNA transcript changes?
What's fascinating here is how structure dictates function. The physical three-dimensional shape of a protein determines how it interacts with other molecules, which downstream regulates the entire transcriptomic network.
Oh, I see.
Right. By feeding DeSCOPE, the structural blueprint of the protein that a specific gene creates, you are giving the network profound contextual clues about the functional role of that gene. The AI isn't just seeing an arbitrary label, it is seeing the mechanistic potential of the gene.
It anchors the prediction in physical reality.
Exactly.
And there is a second mathematical anchor in this architecture, right? The KL divergence loss.
Yes. So computationally, KL divergence measures the statistical distance between two probability distributions.
Meaning it keeps the AI from making wild guesses.
Exactly. Because when you perturb a cell, its fundamental identity doesn't vanish. A modified blood cell is still fundamentally a blood cell. Its transcriptomic distribution just shifts slightly along a manifold.
Right, it doesn't suddenly turn into a bone cell.
No, exactly. So KL divergence calculates the mathematical penalty for deviating too far from the original state. It strictly forces the predicted, perturbed latent space to remain tethered to the control latent space.
It mathematically outlaws those wild, biologically impossible predictions that a heavier, unconstrained transformer model might hallucinate.
Which happens a lot with those massive models.
It really does.
Yeah.
So just to do a quick recap before we move on, we know CRISPR testing is too slow. We know current giant AI models lose to simple math averages because they get lost in the noise. But DeSCOPE fixes this using a zip file CVAE architecture guided by massive protein ID cards from ESM2 and kept realistic by KL divergence.
That's the perfect summary.
So think about this for a second. How would you feel if you spent millions training a giant AI, only to find a basic math average beat it? That's crazy. But let's see how DeSCOPE changes that. Because it is fascinating that using ESM2 protein models and a lightweight architecture allows DeSCOPE to stay so grounded. But that makes me wonder, if it is this efficient at mapping perturbations it already knows, what happens when we throw a complete computational curveball at it?
Like a gene it has never seen before?
Yeah, exactly. A gene it has never encountered in training.
So this is the unseen gene benchmark, and it is a critical test for true generalization. The researchers evaluated DeSCOPE across 5 distinct human cell lines to ensure the results weren't just isolated flukes.
Right, you need a diverse data set.
Definitely. They used K562, which is a leukemia line, RPE1, H1, HepG2, which is a liver cell line, and JERCAT T cells.
And the evaluation metrics they used are quite rigorous. Let's break those down. They looked at overlap at 50 and overlap at 100.
Which is a very intuitive metric.
It is. It asks a straightforward question. Of the top 50 or 100 genes with the most significant expression changes in the real-world lab data, how many did the model correctly identify?
Right. Did it guess the right genes?
Exactly. Then they tracked the Pearson correlation to measure if the AI captured the overall directional trend of the transcriptomic shift. Like, did things go up when they were supposed to go up? And finally, MAE, or mean absolute error, which tracks the exact quantitative deviation in expression magnitude from the ground truth.
And the initial performance data on those metrics is striking, particularly regarding computational efficiency.
Oh, the speed difference is insane.
Right. On the K562 dataset, the massive SCGPT model required 1,464 seconds to train and consumed over 25 gigabytes of GPU memory. DeSCOPE trained in just 40 seconds.
Wow. 40 seconds.
40 seconds, using a mere 1.4 gigabytes of memory. It achieves state-of-the-art accuracy using a fraction of the hardware.
The efficiency is undeniable. But digging into the supplementary data, there is a distinct hurdle, a little bit of a plot twist. When trained and tested on isolated single datasets, DeSCOPE still occasionally struggled to definitively beat the perturbed min baseline on that Pearson correlation metric.
Yeah, this simple average was still putting up a fight.
To bypass this, the researchers implemented a transfer learning strategy called Leave One Out, or LEO.
The LEU strategy is brilliant. It's how you force an AI to learn universal biology rather than over-memorizing the specific quirks of a single dataset.
So how does it work?
Instead of training on just one cell line, DeSCOPE is pre-trained on 4 of the distinct cell lines simultaneously. It maps out the perturbation space across blood, liver, and epithelial cells. Then, it is fine-tuned on the 5th, unseen data set.
I love this part. Computationally, it forces the latent space to calculate a universal translation vector. Let's use an analogy here. It's like, well, if you only ever learn to drive in one highly specific, quiet suburban town, you'll be terrible if you suddenly get dropped into downtown Manhattan.
Right. Oh, absolutely. You'd crash immediately.
Right. You apply those biased, localized rules to everything. But if you learn to drive in 4 completely different chaotic cities, say Tokyo, Rome, New York, and London, you extract the fundamental conserved rules of driving.
Right, you learn to anticipate traffic regardless of the street signs.
Exactly. And that makes you far better at navigating a 5th unseen city. By pre-training across radically different cellular environments, the model extracts the fundamental conserved rules of genetic interaction.
And it worked. Once they applied the LOO strategy, DeSCOPE finally crushed the perturbian baseline consistently across all metrics. It proves that exposing the model to diverse biological contexts is way more valuable than just deepening the parameters.
Okay, so a quick mini recap. They tested DeSCOPE on 5 datasets, found it was incredibly fast to train, but they needed that low strategy training on multiple contexts to really beat those simple baselines.
That LOO strategy really is the secret sauce.
It is. So think about how that applies to dataset curation. If you were designing this AI, which human cell lines would you consider the most crucial to include in your training data to yield the most robust universal knowledge? Because we have figured out how to extract universal rules for unseen genes, provided the AI knows the cellular environment.
Right, but biology isn't neatly compartmentalized. A genetic tweak in a Jurkat T cell does not trigger the same regulatory cascade as that exact same tweak in a HepG2 liver cell.
Which brings us to a massive biological blind spot. How does this architecture handle cross-cell type transfer? Predicting a new gene in the same cell type is hard, but transferring knowledge to a completely unseen cell type.
Yeah, the researchers set up a brutal benchmark here. They attempted a zero-shot transfer.
Which means no peeking at the answers, right?
None at all. They fully trained DeSCOPE on the K562 leukemia cells and then asked it to predict the transcriptomic response of HepG2 liver cells to various perturbations. And they did this without ever allowing the model to see a single perturbed liver cell during training.
So what happened?
It failed completely.
Wait, so the AI completely failed. Why is biology so stubborn?
If we connect this to the bigger picture, it makes perfect sense. In the zero-shot scenario, a non-learning baseline algorithm called delta transfer actually won.
Delta transfer?
Yeah, it's incredibly rudimentary. It simply calculates the exact reaction from the source cell and mathematically copy-pastes it onto the target cell.
Just a straight copy-paste?
Yep. And the fact that this lazy copy-paste method beat the neural network reveals a fundamental biological reality. Cellular regulatory networks are not globally conserved. You cannot port a reaction across tissues because the baseline epigenetic grammar of a liver cell is entirely different from a blood cell.
The AI couldn't decipher the new biological grammar because it didn't have a translation key.
Exactly.
This is where the researchers introduced few-shot learning, specifically 50-shot learning. They provided DeSCOPE with just 50 examples of real perturbed cells in the new target tissue. Just 50.
And the performance jump is staggering. By integrating those 50 perturbed examples, DeSCOPE's Pearson correlation metrics skyrocketed from a baseline of around 0.14 up to an incredible 0.65.
From 0.14 to 0.65, just from 50 examples.
It adapted almost instantly to the new cellular environment.
Let's break down how that actually works in the latent space, because it isn't just memorizing 50 new rules. Those 50 examples act as mathematical anchor points. The CVAE doesn't have to relearn what a cell is from scratch.
Right. It already knows the basics.
It simply looks at how the expression manifold shifted between the source cell line and the new target cell line in those 50 specific instances. It calculates the mathematical delta, the translation vector, and then applies that geometric rotation to the rest of its unseen predictions.
It's elegant math.
It really is. It is the computational equivalent of trying to read a book in a foreign language blindly, which is zero-shot, versus being handed a dictionary with just 50 key words.
50 words, and suddenly the underlying syntax clicks into place.
Exactly. And you can decipher the rest of the text. It highlights the true power of a properly constrained latent space.
It possesses incredible domain adaptation capabilities. It requires minimal data to recalibrate its internal map of a new tissue type.
Okay, another quick recap. Zero-shot learning fails because cell responses aren't perfectly conserved across tissues, which is why delta transfer wins there. But give DeSCOPE just a 50-shot peak, and it adapts rapidly to achieve state-of-the-art results.
The jump is just amazing.
It really makes you think about your own learning style. Like, how much faster do you grasp a complex topic once you're given just a few concrete examples? It changes everything.
It really does. But, we are successfully mapping singular gene knockouts across different tissues now.
Right.
But human pathology is rarely that isolated. Complex diseases, particularly systemic ones like cancer, are usually driven by a network of interacting mutations. We need to look at how DeSCOPE handles combination.
Because it's rarely just one rogue gene.
Exactly. The paper evaluates the model on combinatorial perturbations, tracking 3 specific types of genetic interactions. First is additive.
Meaning 1 + 1 = 2.
Precisely. The transcriptomic effects of knocking out 2 genes independently simply stack on top of each other. Second is synergy.
Where 1 + 1 = 3.
Exactly, where the combined effect creates an emergent response far greater than the sum of its parts. And third is suppression, where the 2 perturbations actively antagonize or cancel each other out. So 1 + 1 = 0.
The latent space math required to predict suppression is fascinating. I know the text highlights a specific suppression case involving the genes RHOX-F2 and SET. When you perturb them simultaneously, they fight for regulatory dominance.
Right. And because DeSCOPE assigns each gene that 5,120-dimensional ESM2 ID card we discussed earlier, its latent space is robust enough to mathematically combine those 2 distinct structural vectors and accurately predict the emergent suppressive outcome.
It correctly predicted the directional change for the top 10 affected genes in this suppressive combination. That completely outperforms models like SCGPT that struggle to synthesize conflicting data signals.
It proves the architecture can handle complex, multi-gene logic. But up until now, we have only been discussing RNA.
Right, the active recipe book.
Exactly. RNA is the output. It is the final transcript. The paper pushes DeSCOPE further by shifting modalities entirely, moving to SCADEC-seq data.
Oh, right. ATAC-seq measures chromatin accessibility. Let's go back to our analogy. If RNA sequencing gives us a snapshot of the active factory floor, the recipe's being cooked. ATAC-seq tells us which blueprints are physically unlocked in the master filing cabinet.
That's a great way to picture it. Our DNA is tightly spooled around histone proteins. If a specific genomic region is tightly wound, it is closed, and transcription factors cannot bind to it.
So the recipes are glued shut.
Yes. If it is unwound, it is open and accessible. ATAC-seq maps out these open regions across the entire genome.
But to process this computationally, the researchers had to filter an unimaginable amount of sparse data. They focus on CCREs, right?
Yeah, cis-regulatory elements, which are essentially the non-coding control switches for our genes.
The primary hurdle with SCADEC data is the sparsity. It has roughly a 90% dropout rate.
90%? That is incredibly noisy data.
It is. Because the assay is trying to capture tiny fragments of open chromatin in a single cell, the vast majority of the data matrix is simply empty. To cut through this, DeSCOPE borrows an algorithm directly from natural language processing called TF-IDF.
Term frequency inverse document frequency.
Right, which is exactly the same math search engines use.
It is a brilliant cross-disciplinary application. Search engines use TF-IDF to determine the relevance of words on a web page. If a word like "the" appears constantly across all pages, its inverse document frequency drops, and it is filtered out as background noise.
But a highly unique word gets prioritized.
Exactly. DeSCOPE treats every individual cell as a document, and every CCRE regulatory switch as a term.
Oh, wow. So by applying TF-IDF, the model mathematically filters out the ubiquitous housekeeping regulatory switches that are open in every single cell. It isolates the highly specific context-dependent switches that actually drive the cell's unique identity.
Yeah, they used the search engine math to narrow the field down to the top 50,000 most critical regulatory elements.
And even dealing with that 90% data dropout, the architecture held up. DeSCOPE achieved 60 to 80% directional accuracy in predicting which specific regulatory regions would be upregulated or downregulated following a perturbation.
Which significantly outperformed EpiAgent, which is a baseline model specifically designed for epigenomic data.
It effectively mapped the downstream structural changes to the DNA's physical accessibility. Okay, so time for our final recap. DeSCOPE handles double gene combos perfectly like synergy and suppression. It translates to epigenomics, the SCADEC data, using that TF-IDF search engine math, and cuts through 90% noise to find the biological truth.
It really is an incredibly versatile framework.
It is. So think about the implications of that dual capability. What do you consider the more critical function for a truly predictive virtual cell? Is it the ability to accurately forecast the final RNA recipes being produced? Or the ability to map the physical opening and closing of the DNA doors that govern the entire system?
Well, you inevitably need both to capture the full regulatory loop, right? A transcriptomic change often triggers a feedback mechanism that alters chromatin accessibility, which in turn alters future transcription.
So what does this all mean for the broader landscape of computational biology? We have looked at how a CVAE avoids the overfitting traps of massive transformers, how protein ID cards inject biological reality, and how mathematical translation vectors allow an AI to jump across entirely different human tissues.
It signals a profound paradigm shift. The current trend in AI is simply to scale up, feed a massive transformer more data, give it more parameters, and just hope it eventually learns biology.
Brute force, basically.
Right. DeSCOPE proves that a lightweight, highly constrained model that respects biological priors is actually far more capable of true domain adaptation. By utilizing structural protein data and enforcing mathematical anchors like KL divergence, you can cross biological modalities and predict unseen variables without needing a supercomputer.
It is a powerful reminder that algorithm design and domain-specific constraints still trump brute force computing. If a localized, efficient framework like DeSCOPE can accurately map the ripple effects of multi-gene combinatorial perturbations across both the transcriptome and the physical epigenome, it accelerates the timeline for personalized medicine dramatically.
Oh, the clinical applications are moving from theoretical to highly practical very quickly.
Imagine the diagnostic protocol a decade from now. You aren't subjected to a gauntlet of physical drug trials to see what works. A clinical AI constructs your unique cellular digital twin. It utilizes models similar to DeSCOPE to simulate millions of targeted, suppressive, and synergistic gene therapies in software right while you wait in the doctor's office.
It's an amazing thought.
It maps the cascading downstream effects across your specific chromatin architecture in seconds, finding the exact molecular intervention required to halt a pathology. We are moving toward a reality where we cure diseases in code before they ever have the chance to physically manifest in your body. Thank you for coming on this dense, highly detailed journey with us today. Keep analyzing the data around you, keep questioning the baseline assumptions, and we will see you on the next deep dive.